Welcome back to Chapter 5, Protein Function. In this lecture, which is part 3 of this chapter, we will examine both qualitative and quantitative aspects of cooperativity. As we saw in part 2, hemoglobin is an oxygen-binding protein that involves structural changes in its multi-subunit, which results as a result of oxygen binding cooperatively. So there are two different models that have been proposed for cooperative binding of a ligand to a multi-subunit protein. The first model, which is a concerted model, says that all subunits of the protein undergo conformational changes and binding simultaneously, which means at the same time. The second model, called as a sequential model, suggests that binding of one ligand to one subunit induces changes in other subunits, thereby increasing their affinity for a ligand. This is much more dramatic. I would consider something like that because when one subunit binds a ligand, the other subunits respond to it by conformationally changing. And as a result of that, another ligand could bind to this conformational, to the subunit that happened to have a conformational change. The concerted model assumes that the subunits of a cooperatively binding protein are functionally identical and that each subunit can exist in at least two conformations and that all subunits undergo the transition from one conformation to other simultaneously. And this is shown here graphically. It can either exist in this conformation shown by circle, or all of them exist in the square conformation. And when a ligand binds, it binds either to a conformation that is circle, and as a result of this binding, all the subunits change to a square. Or when the first ligand binds, the subunits remain in the same conformation and another ligand binds to the next subunit. And that can induce a change when all subunits change or none of them change. This is what it means by undergoing transition from one conformation to another simultaneously, together. In the sequential model, a conformational change in one subunit makes a similar change in an adjacent subunit. And as a result of binding of a second ligand molecule, more likely when the first ligand molecule binds to one subunit. For example, if you assume that all the subunits are in the circle state or the T state, which is a low affinity state, binding of a ligand, which is shown here in the L state, shown as L, and the square. The square is because the binding of one ligand converts T to R state. Now, this can have various different fates. The first one is that this can lead to another conformational change to the subunit next to it. Now, as you notice, it's going from circle to square. Or this specific conformation of the ligand bound state of a protein can go to the next state, wherein another ligand binds to the next subunit. Now this can go via this concerted path, or it can go via this path, or it can go via another path. So this is a sequential model. Now, in this case, not all subunits change conformation simultaneously. That's the difference. There are more potential intermediate states in this model, as you can see. The two models, the concerted model and the sequential model, are not mutually exclusive. The concerted model, as we saw in the previous slide, is viewed as all or none. And it's a limiting case of the sequential model, which means that the sequential model has the concerted model as well. For example, if you look at the T state going from here to here, it can go via concerted model. When ligand binds, none of the subunit changes its conformation, and ligand binds sequentially while the changes in subunits is simultaneous. But sequential model as such is unpredictable because it has various different intermediates. So these are the two different models that are predicted for a cooperative binding of a ligand to a protein. Now, what I need you to take away from these two models is that proteins that uses cooperativity to bind a ligand induces or undergoes conformational changes, and these conformational changes result in further binding of more ligands, and this can happen via multiple mechanisms. I don't want you to remember as to how each and every step happens in the sequential model, but at the same time I want you to understand that these two models do exist, and one can use these mechanisms to explain how proteins function. Cooperativity can be explained using a quantitative description. We have seen this equation, P plus L giving PL. It's a classic protein ligand binding equation. Now, for a protein with n ligand binding sites, this equation becomes P plus NL giving PLN. This means that if a protein has n ligand binding sites, it will bind n ligands. Now, cooperative proteins such as hemoglobins have multiple ligand binding sites, as we have seen. So Ka is equal to PLN divided by P times L to the power n, and hence theta is equal to L to the power n divided by L to the power n plus Kd. And we have seen this equation before, except the power n. And this is what happens for proteins with multiple ligand binding sites. Now let's rearrange this equation. So in this case, let's divide this equation by 1 minus theta on both sides. Now, if you divide by 1 minus theta, what's going to happen is L to the power n divided by L to the power n plus Kd divided by 1 minus L to the power n divided by L to the power n plus Kd, right? Now this becomes L to the power n divided by L to the power n plus Kd divided by L to the power n minus L to the power n plus Kd divided by L to the power n plus Kd, because this term goes here, right? This multiplies with 1. Classic algebra. Now what happens here is L to the power n divided by L to the power n plus Kd divided by L to the power n minus L to the power n cancels out. So Kd divided by L to the power n plus Kd. So this becomes theta divided by 1 minus theta is equal to L to the power n divided by Kd. So for a protein that has n ligand binding site, this equation now becomes theta divided by 1 minus theta is equal to L to the power n divided by Kd. One of the very reasons why we divided by 1 minus theta is to simplify this equation. Now, taking log on both sides, we get log theta divided by 1 minus theta is equal to n log L minus log Kd. And this is called as the Hill equation. And one of the important parameters of a Hill equation is n. That is the Hill coefficient, or it dictates the degree of cooperativity. Remember this point. Please remember this equation, that is log 1 theta over 1 minus theta is equal to n log L minus log Kd. And also, what happens when n changes? When n is equal to 1, there is no cooperativity. Whereas when n is greater than 1, there is positive cooperativity. And when n is less than 1, there is negative cooperativity. Important points. Please remember this. So the Hill equation can be written in a logarithmic form, right? And this is what it is. We can plot log theta over 1 minus theta versus log L. So you can consider is it in this fashion. Y is equal to mx plus c. Right? If you plot y, which is log theta on 1 minus theta versus x, you will get a straight line. Right? Hill plot allows us to determine cooperativity. The slope of a Hill plot is the degree of interaction, and the slope of a Hill plot is also known as the Hill coefficient. Now if you go to this website, it gives an interactive description of Hill plots. As you can see from this plot, as you change the value of n, the slope changes. And if you plot log theta on log theta over 1 minus theta versus log L, you get straight lines. And you can obtain meaningful information from this plot. The intercept, y-intercept, is given by Kd. The slope is given by NH. And this is how you obtain these two parameters, because these are the two important parameters that you need. Slope, or NH, indicates how many ligand binding sites are there in a protein. Kd indicates how strong an interaction does a protein have with a ligand. Cooperativity is a special case of allosteric regulation. What is an allosteric protein? In case of an allosteric protein, binding of a ligand to one site affects the binding properties of a different site on the same protein. Right? We briefly talked about this. in the previous lecture. For example, if a protein binds to a ligand, right, and this affects the binding of this protein to this ligand at a different site, which means that this can change the conformation of the protein and it can bind to another ligand on a different site, right? That is allosteric or allosteric protein. Can be positive or negative. Homotropic is a special case of allosteric, in which the normal ligand of the protein is the allosteric regulator. This is the case. The normal ligand binds, changes the conformation, and as a result, protein can bind another ligand. The second case is heterotropic. A different ligand affects binding of the normal ligand. For example, if this is the protein, a different ligand can bind in this site, and this can affect the normal binding of this protein. And this can change the conformation of the protein. This is the ligand bound, and it can make the protein to bind to its actual ligand. And this is called as heterotropic. A third one, so a cooperative protein has a positive homotropic regulation, or cooperativity is a special case of positive homotropic regulation. In addition to carrying nearly all the oxygen required by cells from the lungs to the tissues, hemoglobin carries two end products of cellular respiration. They are H+ and CO2 from the tissues to the lungs and the kidneys, where they are excreted. CO2, or carbon dioxide, produced by oxidation of organic fuels in mitochondria, is hydrated to form bicarbonate. As you can see, CO2 plus H2O giving HCO3- plus H+. This reaction, as you can see, is catalyzed by the enzyme carbonic anhydrase. This enzyme is particularly abundant in red blood cells. Carbon dioxide is not very soluble in aqueous solutions, and bubbles of carbon dioxide would form in the tissues and blood if it were not converted to bicarbonate. As you can see, from the reaction catalyzed by carbonic anhydrase, that is carbon dioxide plus water giving bicarbonate and H+, every carbon dioxide, that is every molecule of carbon dioxide that gets converted to HCO3- result in the formation of a molecule of H+. So, as the concentration of H+ increases, pH decreases in the tissues. The binding of oxygen by hemoglobin is profoundly influenced by pH and carbon dioxide concentration. The binding affinity of hemoglobin for oxygen depends on the pH. H+ binds to hemoglobin and stabilizes the T state. Do you know what T state is? Is the low affinity state for oxygen. The way it does is by protonating a histidine, His 146, which then forms a salt bridge with aspartate 94. This leads to the release of oxygen in the tissues. So, the point is, as the pH decreases, the binding affinity of hemoglobin for oxygen decreases. The pH difference between lungs and metabolic tissues increases efficiency of the oxygen transport. This is known as the Bohr effect. the effect of pH and carbon dioxide concentration on the binding and release of oxygen by hemoglobin. That is Bohr effect. It's very important to understand this effect. Again, I rephrase. If you increase proton concentration or H+ concentration, you are decreasing the affinity of hemoglobin to oxygen. This can also be explained by this plot, right? As you can see, if you plot Y, which is the fraction of bound ligand versus PO2, and as you could see, increase or decrease in pH decreases the affinity. This is pH 7.6, and this curve, as you can see, shift towards the right, which means the KD increases, right? So this explains as to how, when the concentration of oxygen increases in the lungs, hemoglobin binds oxygen and releases protons. But when the concentration is low, as in peripheral tissues, protons are bound and oxygen is released. So proton concentration is very high in tissues, and hence oxygen is released. This is how the interplay between pH and oxygen affinity helps us to understand how hemoglobin binds oxygen in the lungs and releases it in the tissues. Hemoglobin is also involved in carbon dioxide export. Carbon dioxide is produced by metabolism in tissues and must be exported. 15 to 20% of carbon dioxide is exported in the form of carbamate on the amino terminal residues of each of the polypeptide subunits in hemoglobin. For example, hemoglobin also binds carbon dioxide, again in a manner inversely related to the binding of oxygen. Carbon dioxide binds as a carbamate group to the alpha amino group at the amino terminal end of each globin chain, forming carb-amino hemoglobin. This reaction produces H+, as you can see, and this contributes to Bohr effect. The bound carbamates also form additional salt bridges, and this stabilizes the T state. Now remember, one of the interaction was the histidine that gets protonated, and that interacts with an aspartate, forming a salt bridge. In addition to that interaction, this carbamate that is formed at the N terminus could also form salt bridges to stabilize the T state. 2,3-bisphosphoglycerate regulates oxygen binding. 2,3-bisphosphoglycerate is a negative heterotropic regulator of hemoglobin function. Wow, a negative heterotropic regulator, which means that this binding of this ligand, the ligand is 2,3-bisphosphoglycerate, when that binds to hemoglobin, it makes hemoglobin to bind more oxygen. Now, negative means that when this ligand binds to hemoglobin, it makes hemoglobin to bind less oxygen. That's what it is. And this is 2,3-bisphosphoglycerate. This is present at millimolar concentrations in erythrocytes or red blood cells, and is produced from an intermediate in glycolysis. Small negatively charged molecule. That's what this ligand is, 2,3-bisphosphoglycerate, which means two phosphates. It's a small molecule, but it's negatively charged. 4 5 negative charges, 1, 2, 3, 4, 5, right? And this binds to the positively charged central cavity of hemoglobin. It stabilizes the T states, which means low affinity for oxygen. Oxygen hemoglobin dissociation curve is explained really nicely in this video. I suggest you watch this video. It gives a really nice quantitative description of the graphs as to how hemoglobin binding to oxygen changes. Sickle cell anemia is a hereditary human disease which is caused due to a mutation in hemoglobin. Normal hemoglobin binds oxygen reversibly, as we know. Sickle cell hemoglobin, when it is deoxygenated, it becomes insoluble and forms fibrils in the red blood cells, causing deformation, as you can see on the right here. This is a normal hemoglobin, and this is deoxygenated hemoglobin S, just formed fibrils. This is caused by a single amino acid mutation in hemoglobin, and that is the cause of this hereditary disease. Obviously, there is a genetic mutation which results in the mutation of a single amino acid in hemoglobin, and that mutation is glutamate to valine. Remember, glutamate is a negatively charged amino acid versus valine is a hydrophobic amino acid. Valine creates a sticky hydrophobic contact with other hydrophobic residues, resulting in insoluble aggregates. So these are the insoluble aggregates that I'm talking about. Subtle differences between the conformations of hemoglobin A and hemoglobin S results from a single amino acid change in the beta chains. So if you look at hemoglobin A and S, subtle differences in the beta chain, which is a result of one amino acid change. This makes hemoglobin S sticky, and it sticks to other hemoglobin S molecules and forms these strands, resulting in the formation of a fiber. This results in the formation of this disease. As a result of this change, deoxyhemoglobin S has a hydrophobic patch on its surface, which causes the molecules to aggregate into strands that align into insoluble fibers. Now remember, sickle cell anemia is a life-threatening and painful disease. People with this disease suffer repeated crises brought on by physical exertion. Without medical treatment, people with sickle cell anemia usually die in childhood.